cytotoxicity and genotoxicity. Because our customers are so important to us, we make sure to test if our products and ingredients are safe. We do a special test that shows any damage to cells and DNA. Cytotoxicity and genotoxicity represent the degree of toxicity of a particular substance. We perform these tests by seeding blood into cell culture media. We incubate our media with the human blood cells for 72 hours. After incubation, we add a hypotonic solution and a fixative to help disperse the chromosomes so we can see them better while analyzing them. By observing any changes in cell number or in the structures of chromosomes, we can know the exact cytotoxicity and genotoxicity of a certain substance.
Antibacterial testing is done using the agar-well method. We place pathogens, which are microbial bad guys, onto the agar inside the petri dishes. We then make little wells inside the agar and insert the probiotics into the wells. We place our petri dishes in the incubator for about 24 hours. After incubation, the newly formed zones of inhibition indicate the probiotics' antibacterial potential. A zone of inhibition represents no bacterial growth on the agar. It is a clear zone with no pathogens in it. This is important because we want the good bacteria to naturally kill any bad bacteria that are present in the gut.
The Universal Protease Assay. In the process of creating Masszymes 4.0, we needed to test the enzyme activity of various proteolytic enzymes individually, as well as synergistic blends of proteases. To do this, we use the Universal Protease Assay. Proteases are the enzymes that break down protein, so we needed to measure and compare their activity to determine the maximum protein digesting capability. We performed the Universal Protease Assay by mixing the tested enzymes with proteins commonly found in food, such as casein, gluten, or whey. Now, once the enzyme is mixed with the protein will be broken down by the enzyme into individual amino acids. One of these individual amino acids is tyrosine, which reacts with the reagent called folin. Now, when this happens, the liquid turns blue, and the intensity of the blue color is measured on a spectrophotometer. The deeper the blue, the more effective the enzyme blend.
Antibiotic susceptibility testing. The aim of this test is to determine what probiotics can be taken while on an antibiotic regime. To test this, we place the probiotics on agar plates and afterwards apply an antibiotic disc on the same plate. After a 24-hour incubation, we determine if zones of inhibition appear around certain antibiotics. If a probiotic does not have a clear zone of inhibition, we know that it is a good bacteria to take while on an antibiotic regime.
Research-Backed testing. Research is an indicator of cellular metabolic activity. It is based on enzymatic reduction of an indicator dye. Living cells are metabolically active and are able to reduce research to the strongly fluorescent dye resorufin. The fluorescent output is proportional to the number of viable cells.
Tissue culture plate method. Healthy biofilms are very important for our health. This is why we use tests to evaluate the biofilm forming capacity of our probiotics. This test is called the tissue culture plate method. We put good bacteria into plastic wells and allow them to grow for 24 hours. After that, we spill the free-floating bacteria and evaluate the amount of biofilm formed. This is done by staining the wells with crystal violet. Biofilm formation is detected by the presence of intense purple color, and the results are read using an ELISA plate reader.
the Bradford method. We use the Bradford assay as a simple and accurate way to figure out how much protein is present in a solution. The Bradford method is based on a color change. When the dye reacts with amino acids, the solution changes color. This change in color is directly proportional to the protein content of a solution.
Tube test for biofilm formation. A healthy biofilm is very important for our health. Considering this, we use tests to evaluate the biofilm forming capacity of our probiotics. This test is called the tube test method. We put good bacteria into plastic tubes and allow them to grow for 24 hours. After that, we spill the free-floating bacteria and evaluate the amount of biofilm formed. This is done by staining the tubes with crystal violet. Biofilm formation is detected by the presence of intense purple color, and the results are examined visually.
DPPH. The DPPH free radical assay is a test we perform to analyze the antioxidant activity of different substances. DPPH is a stable free radical molecule. Now, free radicals are toxic to humans, so we perform these tests to see how high a certain substance's antioxidant activity is, because antioxidants protect the body from free radicals. In this method, we observe the electron transfer that occurs when an antioxidant converts the free radical DPPH to a more stable form. DPPH itself is a deep violet color, and when the electron transfer occurs, the liquid turns a pale yellow, indicating antioxidant activity. We measure how intense the color change is on the spectrophotometer, and we do the experiment to find the amount of substance we need to reduce DPPH by 50%.
Bile salt endurance test. For probiotics to reach our gut, they must survive the bile salts in the small intestine. Because of this, we perform bile salt endurance tests. Each probiotic is added to different bile salt concentrations and allowed to grow for 24 hours. The probiotics are then placed on special agar at different time periods in order to see which probiotics are able to survive the longest.
The ABTS assay. The ABTS assay is one of the most common antioxidant assays for biochemical tests. ABTS is a free radical acting compound that has a very intense blue-green color when it is in its radical cation form. So, in order to test the efficacy of a certain antioxidant, we mix it with ABTS. If the antioxidant is effective, it will convert the ABTS back into its neutral form by donating hydrogen atoms, and the solution will turn colorless. We measure the color change on the spectrophotometer at 734 nanometers. After doing this test at several different concentrations, we generate a trend line. We can then use this to find what amount of antioxidant substance will inhibit the ABTS free radical by 50%. This will tell us if we have a potent antioxidant on our hands.
Acid survival test. For probiotics to reach our gut, they must survive the acidic pH in the stomach. Because of this, we perform the acid survival test. Each probiotic is added to media of different pH values and allowed to grow for 24 hours. The probiotics are then placed on special agar at different time periods in order to see which probiotics are able to survive the longest.
Plant assays. As with our stellar probiotic blends, we're creating plant formulas to boost your health and well-being. We blast a plant with the stressor and then analyze the direct changes in the chemicals that they produce. These experiments and analyses are paving the path for some of the most effective plant-based products on the market.